Thanks for the invitation today. My name is Bryce Chackerian. I'm a professor in the Department of Molecular Genetics and Microbiology at the UNM School of Medicine. And my group works on vaccines. And today I'm going to talk to you about our efforts to develop a couple of different vaccines for mosquito-borne diseases. So I just wanted to start with some of my collaborators and some of our funding for this project. So this is a project that we've been working on at UNM in collaboration with a number of other groups there, including Pavan Mutil, who's in the School of Pharmacy, and Justin Baca, who's an emergency medicine physician. And we've been collaborating with a number of other institutions. So for our malaria work, we've been working with Fidel Zavala, who is real pioneer in the development of animal models to study malaria vaccines and other malaria interventions. We've been working with Sunil David, who's developed a number of very potent adjuvants. These are substances that can increase the potency of vaccines, and his company is called ViraVax. And then for our efforts to develop vaccines to prevent transmission of viruses from mosquitoes to humans, we've been working with Angela Bosco-Louth. She's in the School of Veterinary Medicine at Colorado State University. So the work that I'm going to talk about today has been, the malaria work has been funded by an NIH grant. That grant, we're in the second year of a 5-year grant, and that's giving us a total of about a little over $3 million of funding to develop the malaria vaccine. And then we're also so we've developed a vaccine platform technology. That platform is based on something called a virus-like particle. And as the name implies, a virus-like particle is something that looks like a virus, but it isn't infectious. So it's essentially just the protein shell of a virus particle, and it lacks the viral genome, so it can't infect people. So these virus-like particles, it turns out, are very, very immunogenic. They can induce very strong immune responses, and that's for a number of reasons, but basically the immune system has developed a number of different mechanisms to recognize these structures as dangerous. And by doing that, the immune system induces very strong immune responses against it. So what we can do then is that we can take advantage of those properties, take the virus-like particle, and then use it as a vaccine platform system. And what I'm showing here is a very simple technique that we do, where we start with the virus-like particle, we modify it with a target that we're interested in developing a vaccine against. We display that target in a highly multivalent, dense array on the surface of the virus-like particle. And again, that confers strong immunogenicity to the target, and we can get strong antibody responses, we can get very long-lived antibody responses, and we can get those responses with very low doses of the vaccine. So it's a very powerful technique. This has been referred to by others as essentially a sheep in wolf's clothing, okay? So this is, again, a safe technology, but again, to the immune system, it looks dangerous. So the question was, can we utilize this technology to develop highly potent vaccines for mosquito-borne diseases? So as I mentioned, as I alluded to, the sort of two general classes of targets that we're interested in are viral-borne diseases burden of disease worldwide, mostly dengue virus, but these other viruses contribute as well. So in 2020, there were over 400 million cases of viral infection from these arboviruses, resulting in over 700,000 deaths. So the second class of vaccines that we're interested in are vaccines for malaria. These are carried by another mosquito called the Anopheles mosquito. And malaria is caused by a parasite called Plasmodium falciparum. And again, there's a huge burden of disease caused by malaria. So 240 million cases, over 600,000 deaths, mostly in sub-Saharan Africa. So again, two classes of pathogens that cause a huge amount of mortality and morbidity worldwide. So, so talking a little bit about sort of what is the scope of interventions for these different classes of pathogens. There are 2 malaria vaccines that have been approved by the World Health Organization recently in just the last couple of years. They're approved for use in Africa, but their efficacy, unfortunately, is poor. Okay, so the first vaccine has an efficacy around 30%. The second vaccine seems to be better, but also not optimal. And another feature of these vaccines is that protection seems to be fairly short-lived. For arboviruses, there's really only one effective vaccine. That's the yellow fever vaccine that was approved almost 90 years ago. And there really aren't any effective interventions that target mosquito factors that enhance transmission. And that's one of our goals, and I'll get to that in a second. So, I mentioned before that these exact a huge burden worldwide, but because of climate change, this is a burden that is expanding. Okay, so these are a couple of articles in the New York Times. Temperatures rise, these mosquitoes expand their range further northward. That's also going to lead to an expansion of these diseases, outside of tropical and subtropical areas. And for those of you who live in Albuquerque and have seen in the last couple of years, just the number of mosquitoes that we now have in the summertime, I think that's pretty obvious that that is happening here as well. Okay, so now I'm going to talk about the 2 vaccines separately. First, I'm going to talk a little bit about the malaria vaccine, which is further developed. So just very briefly, malaria has a very complicated life cycle, and that's one of the reasons why it's been very difficult to develop malaria vaccines. So this is a parasite that, again, is carried by these Anopheles mosquitoes. When the mosquito bites you, the parasite goes to your liver, and then in the liver, it propagates, it differentiates, and moves into the bloodstream. In the bloodstream, that's where you get the clinical signs of malaria, anemia, and more serious consequences. And then the parasite changes again, goes back into the bloodstream, and goes back into a mosquito host, and this cycle repeats. And so this is a parasite that's constantly changing its form. And again, that has complicated efforts to develop vaccines that can target things that are on the parasite constantly. And the most effective vaccines are thought to be ones that prevent that very first step, the liver step. And one of the problems with that is that if even a single parasite gets into the liver, that can seed the blood stage. And so a vaccine that is effectively able to block infection by malaria needs to provide what's called sterilizing immunity. It needs to prevent every single parasite from getting to the liver. And that's a huge burden for vaccines. You have to induce very strong antibody responses, and those antibody responses have to be long-lived because you can't rely on a memory called circumsporozoite protein. This is a protein that's on the outside of the parasite, and it's involved in the parasite moving to the liver and infecting liver cells. The vaccines that I mentioned before that have been approved target CSP, but again, they don't work very well. And so our innovation is that we've identified a particularly vulnerable target on the CSP protein. This is a target on the protein that seems to be particularly susceptible to antibody responses. And so our goal then was to develop a vaccine that targeted this essentially Achilles heel of the parasite and see if that could improve efficacy. So this is just a little bit of data that sort of led to that. So on the left, what I'm showing are clinical trials of a monoclonal antibody that targets that vulnerable region. This was a study that was done by the NIH where they infected volunteers with malaria and looked to see whether treating them with the antibody could prevent infection. And suffice it to say, they showed that that was the case. And there are now studies in Africa where they're looking at the ability of that monoclonal to block infection. And so what we did was we basically took that information and we designed what's called a virus-like particle-based vaccine targeting that short region of the CSP protein. So basically what we did was we used a chemical crosslinker to array that target on the surface of the virus-like particle at high density so that we can get strong immune responses. This is a picture of what that vaccine looks like. And we've published a couple of papers on this vaccine in Nature journals in the last couple of years. And so just to show you sort of the high-level results, we then moved to a mouse model of malaria infection, and we looked at the ability of the vaccine to protect mice from infection by Plasmodium. So you might think, so what we showed here is their vaccine. We first looked at the ability of the vaccine to prevent the parasite from getting into the liver, and so that's what's shown here on the left. The green dots are our vaccine. The pink dots are the clinically approved GSK vaccine, which is called RTSS. And you can see that both vaccines are very effective at preventing parasite in the liver. But the really key is, can we prevent the parasite moving from the liver and getting into the bloodstream? Can we provide that sterilizing immunity that I mentioned before? And that's what we're showing on the left. So our vaccine was able to prevent blood stage disease in 80% of the animals that we vaccinated. RTSS, this vaccine that's clinically approved, was 40% efficacious, which is similar to its efficacy in humans. So again, our vaccine seems to be superior here, at least in this mouse model, to the current approved vaccine. Okay, and then in the last couple of minutes, I want to talk a little bit about our efforts to develop vaccines to prevent viral infection. So when a mosquito takes a blood meal, obviously it needs to get to the blood. And one of the things that mosquitoes do when they take a blood meal is they actually inject a number of different proteins into your skin that lead to your vasculature to expand so they can access the blood. And so this is a very common feature of mosquitoes. But it turns out amongst those proteins is something that's called sialokinin. And what sialokinin does is it does exactly what I said, but that process also enhances the ability of viruses to infect, to move into the bloodstream, which is an important first step in viral infection. And this is a common feature for all the arboviruses that I mentioned before. Sialokinin in mosquito saliva enhances infection. So that led us to hypothesize that we could potentially right now, but we've developed vaccines targeting sialokinin. This is just data where we've looked at the efficacy of these vaccines over a year after immunization. And all I want to show you here is the 2 candidate vaccines that we've developed. They induce strong antibody responses, and those antibody responses are lasting the life of the mice. And again, that's important. We need long-lived antibody responses to prevent this peptide from doing the things that allow viruses to infect more readily. So again, our current status for the malaria study, we have a $3 million grant from the NIH. We've developed a candidate vaccine that provides stronger protection than RTSS. We're evaluating some vaccine refinements, and we're hoping to move with some support to move into the pre-IND steps, so manufacturing and toxicity. And there's some data on this platform. This platform has moved into clinical trials before, so we think that's very a feasible goal. For the second project, we've developed these candidate vaccines targeting this mosquito salivary protein. We've identified a partner at Colorado State who's going to be able to look at whether these vaccines can prevent or decrease the efficiency of viral infection. And as I mentioned, an NIH grant to fund these studies is pending. This is my email address, and I'd be happy to answer any questions.
Born and growing up in India and being bitten by mosquitoes, this is welcome news to me. I was looking up on the VLP platform. I have heard this some time back, and I was like thinking where I have. It seems that a team of scientists based So in terms of the patent protection from an international scene standpoint, how does this have any kind of conflict?
That might be a better question for Greg, actually. But I can tell you that, so I've been working on these sorts of platforms since the mid 1990s. Okay, so I have a lot of experience, and one thing that's been very fulfilling for me is the recognition that this sort of strategy for developing vaccines is so effective. So there is now a lot of competition in people who are using virus-like particles as a way of improving efficacy of vaccines. So this particular platform, we were the first to develop this specific vaccine, again, targeting this specific vulnerable epitope on this protein, CSP. There are other groups that have tried displaying larger domains of this protein, using virus-like particles, not using virus-like particles, and they've shown some positive results, including maybe the one that you discovered by Googling it. But I think our vaccine is unique in that we are targeting this vulnerable epitope. And again, that is critical for malaria because you not only need these strong responses, but you need to find things that are actually vulnerable to those strong responses. So hopefully that answered your question.
Yeah, it does. But given the recent love story between Massimo and Apple, I would still recommend someone has to look into it. Thank you.
Yeah, great.
No, thank you so much for this presentation and for the discovery and the work put into this. As we start thinking about the next steps that you were talking about of manufacturing and putting this out into the market space there and beginning the preliminary thought the grants into kind of the commercialization process.
Yeah. I mean, there's a bunch of questions, I think, in that question. So in terms of, pre-IND, that's obviously a focus. This is something we're going to have to, deal with regardless of what the market is. And I think we have a pretty good handle on that. As I mentioned, we're using what's called a bacteriophage virus-like particle. So this is a platform that has gone into clinical trials before. So there's a lot of information on how to make this particular platform. And there's also good safety and immunogenicity data in humans. So that gives us a lot of confidence that these vaccines are going to be effective and safe in human populations. Now, in terms of the market potential, that is a whole other story. So when we're talking about malaria and we're talking about some of these, again, mosquito-borne diseases, these are things that are mostly affecting the developing world, okay? And, that's a noble goal to develop vaccines for the developing world, but sometimes it might not be a lucrative one. And so, where do we see our market? We see our market as governments, okay? And, that makes a lot of sense. And not just, the governments of developing world and non-governmental organizations that can provide the vaccine, but also the US government, who has troops overseas that are in areas that are susceptible to these sorts of infections. So that would be, probably the largest market. But then, as I mentioned, it's really hard to predict whether these diseases are going to spread north. And as they spread, and I think it's inevitable that that's going to happen. And of course, we have mosquito control and all those things that are going to help. But for travelers that are going to these places, and potentially in Europe and the United States, we may need these vaccines as well. So that's also a future market.
At what point do you go to places like the Gates Foundation? Is that later on when you're ready to do field trials?
Yeah, so we're already collaborating with PATH right now, which is sort of funded by Gates. And so they were the ones who allowed us to do the side-by-side comparison of the GSK vaccine with our vaccine. So we've opened those channels. But that, Gates has been a great supporter of both RTSS, but also R21, which is the second malaria vaccine. And so, that's absolutely a potential, way that we can move this forward.
Exciting technology.
Thanks.